everyone. We are going to see about their structure and classification morphology of microbes. So from this unit 1, I am Sornambiga, SNS College of Nursing, Assistant Professor. So we are going to discuss about what is prokaryote, eukaryote, viruses, beyond this, to that how they are structured, well structured, organized, and what are the classifications are there, and how we are animated visuals are there in this video. We will watch on this. Here, again, that's structure of prokaryotic cell. So everywhere in the layer of packed surveillance bunch without the eukaryote frills to enrich the polysaccharides to capsule foil that immune system. So in that last video we saw that polysaccharides give the structure of bacteria. Similar that the Streptococcus pneumoniae to clogs the lung slobs and the gram-positive saw that causing that more infection comparatively gram-negative microorganism, that exotoxins. That phospholipids is a bilayer to that ribosomal DNA and that antibiotic resistant to the traits of the bacteria present with that flagellas, pili, fimbriae, conjugation or innovation processes and to inclusion of the stores into that to hygiene practices to handle with that care of these prokaryotic cells or more virulent in these cases. Next, we are moving on to that eukaryotic cells. Look at the eukaryotic cell compared with prokaryote. It's completely different to the fungals and protozoal spores. So in that chitins or cellulose or the cell wall to grips many like the candida barriers to the thresh of microorganisms to the plasma membrane. In this case, to steroid that stand for the antifungal targets to oversess selectively to increase that ribosome. So that harvests chromatin own to the DNA from that fateful division from the mitochondria to that ATP to ADP to convert their own food by the way of these cells. That eukaryote having own nucleus and prokaryote is not like that. So they have nucleus, nuclear membrane, Golgi apparatus, mitochondria, but prokaryotic cells are not like that. They don't have that powerhouse of the cells also. In that amoeba, cilias, wipe that paramecia, and that we have to spotting of that opportunistic blooms of diabetes also. Virus structures. Viruses are enveloped influenza bacteriophages. Whether it is that bacteria combined with virus or bacteria infected by the virus is called bacteriophages. The best example is T4 bacteriophage. It is easily affected from that. After that, we are crossing more probably what are the structures or virus. Almost we saw all structures. In this video, we saw simple virus structure in the DNA, whether it is RNA segmented, handled with the capsid, the assembly of the protein, to the icosahedral head. After that, to enveloped in the lipid head of the host, spiked with hemoglobin, to the airway bubbles to that grip into the spot, to the 120 glycoprotein, to the HIV and CD4 visit in the cases. In the snake sheath, basoplates, and that base platters and fibrous eject of DNAs. These are there in that bacteriophage structures. And it has that nosocomial infections also to spread one person to another person. And microbial uh, classifications of the tree. In the classification tree is very easy to understand what are the bacterial category, archaea category and eukaryote category. In that bacteria category, we saw spirochetes, proteobacter, uh, cyanobacters, and plantobacters, plantomyces, and aquasis, and some other things also we have able to see from this slide. So in the classification branches is a taxonomy to navigate the realms. The five roots of that cellularity to domain that phyla to the foreign substance of the cells to elaborate the prokaryotic cells difference between the eukaryotic cells also. Here again we saw how the cocci, spirillars, bacilli and other cells are present in this nature. Here that one of the important thing is size of the bacteria and size of the virus and size of the other microorganisms comparatively. But we cannot able to see anything from this our naked eye. We can able to see from the way of microscopes also. So it is a one important thing we have to know how that microorganisms very small comparatively in that nature. It is a colonization formation. How they are colony. Once that microorganism wants to attach with that human host, after only they can able to produce and cause the infections. So they are first attaching to the human host. After that, it is multiple by the way of binary fissions or as a budding formation. After this, it is a developmental into the next stage. It is maturing the other another way to development. After that, it is maturing to the baby cells to adult cells. After that, it is actively dispersed and spread the disease to the another process also. This formation is completely called as the biofilm formation. So attachment, colonization, development, mature and active dispersal. So the complete one process will take within 24 hours. This process is called biofilm formation of the cells. So one of the most important thing after the biofilm, how much time will take and how they are growing. So this is the growth curve, one of the important thing from the microbiological strategies. It is the four phases are there. One is lab phase, that another one is exponential growth phase, stationary phase, death phase. In this lag phase, there are one small number of microorganisms only can able to produce their enzymes, and in that lag phase it is doubling by the way of binary fission within 20 minutes. So in the generation time they are producing by the way of lag phase to log phase. After the stationary phase, they are not reducing condition, they are not death condition also. They are stable in the same category of condition. After the death phase, it will be numberly wise, it is slow their growth and it is going to death of the stages. And classification of microbes by the way of nutrition, it is autotroph and heterotrophs. That autotrophic is using the light energy and chemotrophs also using their chemical energy. In the heterotrophs also there it is a saprophytic and parasitic. They are taking their nutrition from the live cells or as a dead cells from the other cells also. Here how that microbial nutrition growth indication, I will give the small chart for your reference purposes. Here this video will be more useful for your learning purpose. Thank you.